Hello, everybody, and welcome to the College of Remote and Offshore Medicine and Academy of Wilderness Medicine, Wilderness Medical Society student interest group. And as part of our lecture series, we will be delivering a presentation on bioterrorism. My name is Tim Cranton. I'm teaching faculty and a professional practice advisor for the College of Remote and Offshore Medicine. I'm also an ex-British Army combat medic with chemical, biological, radiological, and nuclear incident training. I'm a NATO Special Operations medical instructor, also instructor for tactical emergency care, all hazards disaster response, as well as pre-hospital trauma life support, and member of the NAEMT as affiliate faculty. I'm currently registered as a paramedic in the UK, as well as an offshore medic. I was also in the UK Fire and Rescue Service, and again, trained in chemical, biological, radiological, and nuclear response. And I was part of the response team for the Greater London area for the New Dimension project. This was brought about after the September 11 attacks in London. I'm a remote healthcare practitioner, as already mentioned, as well as dive medic, and a director of paramedic education, who are a training organization in the Czech Republic, which is where I live. And as I mentioned, I'm a member of COROM. So the picture we see here is atypical of one of the duties that I performed when I was in the UK Fire Service as part of the New Dimension concept. And we provided, or part of our role for the Greater London area was to provide emergency mass decontamination for a large number of the local population. So we're not talking 2 or 3 people, we're talking hundreds and thousands of people. So today's presentation, I'm going to look at the definition of bioterrorism. I'm going to go through a short history, or a recent short history, and we're going to look at the ideal biological weapon. We'll look at epidemiological clues to an event. We'll look at biosafety levels, as well as some individual biological agents. I'm going to go through the categories of what we describe as critical biological agents, describe more of bioweapons, look at some biological research and give you some information on that. But one of the things that's most important really to us as emergency medical responders is how we would manage a bioterrorist type incident. I'll summarize that, and at the end I've provided you some resources that you can take away and you can do some self-learning with. So by definition, bioterrorism is the deliberate and illegitimate use of biological agents, such as bacteria, viruses, toxins, or other biological substances, to threaten or harm individuals, communities, or nations. Bioterrorism can take many forms, including the release of dangerous pathogens, contamination of food or water supplies, or the dissemination of harmful biological materials to cause widespread harm and panic. It's considered a serious threat to public health and national security worldwide, and efforts are made to prevent and respond to potential bioterrorist attacks. These biological agents are typically used with the intent to cause fear, illness, or death among humans, animals, or plants as a form of terrorism. Now, bioterrorist incidents are relatively rare and are typically characterized by deliberate acts of using biological agents to cause harm, illness, or fear. And we should also be aware that any victim of a poisoning or exposure to a substance becomes themselves a biohazard to any responder. What I've got now are some examples of a few notable bioterrorist incidents that have occurred worldwide in the past. So we have an organization called Shinrikyo that were a Japanese cult, And in the 1990s, they attempted several bioterrorist attacks. One of the most significant incidents involved the release of the deadly nerve agent sarin in the Tokyo subway system in 1995, which resulted in 13 deaths and thousands of injuries. This cult also attempted to disseminate anthrax, botulinum toxin, and other biological agents. If we look at the slide, you can see several photographs. And we've got a guy there who's got self-contained breathing apparatus on and quite a high level of personal protective equipment. But that can't be said about people that are close to him. And this is what we end up with in, typically any kind of incident like this, where we can have responders that may have access to a level of personal protective equipment that protects them, but other people do not have that luxury. Sometimes even responders themselves don't have a level of PPE that may be required to protect them. And again, as we can see, we have a large number of people that have been affected here. In 2001, we had the anthrax attacks in the US. This was in the wake of September 11 terrorist attacks, and letters containing anthrax spores were sent to media outlets and politicians. These attacks resulted in 5 deaths and several illnesses, and the perpetrator was not definitively identified, but is considered a significant bioterrorism incident. So the bacterium, so this is Bacillus anthracis, it produces dormant spores, so they can lie inactive for an extremely long time in soil, and they can live for decades. But when these spores get into the body of an animal or a person, they can be activated, so they turn into active growing cells. When they become active, they multiply rapidly, they spread to parts of the body, they produce toxins, and they can cause severe illness and death. Members of the Renishi cult in Oregon in 1984 contaminated salad bars in local restaurants with salmonella in an attempt to influence a local election, and this resulted in hundreds of cases of food poisoning. Now, Salmonella enterica typhi, we've got an example of that here in the slide that's on the right-hand side. It's a gram-negative bacterium that's responsible for typhoid fever and has been a burden on developing nations for generations. But despite significant efforts in research and medical advancements, typhoid fever is still a major worldwide public health concern. Sverdlovsk anthrax outbreak, 1979 in the Soviet Union. This incident involved an accidental release of anthrax spores from a Soviet biological weapons facility. So here we have an instance of arguably legal production, or certainly retention or storage of biochemical weapons, or certainly substances that could most certainly be weaponized. In this case, at least 68 people died as a result of the outbreak. A lot of those would have gone unreported. Now, anthrax makes a good weapon because it can be released quietly and without anybody knowing. The microscopic spores could be put into powders, sprays, food, and water. And because they're so small, you might not be able to see them, smell them, or taste them. Anthrax has been used as a weapon before. So it's an infection caused by the bacterium Bacillus anthracis, and it can occur in 4 forms. So skin, lungs, intestinal, and injection. In Salisbury, in the United Kingdom, in March of 2018, we had Novichok poisoning. So the UK Novichok poisoning incidents refer to a series of poisonings involving the use of Novichok nerve agents, highly toxic chemical substances developed during the Soviet era, but on British soil. Here are the key details. And we can see here with the map on the right-hand side, the route that the people that were exposed to the poisoning took. And this is how we can see or have an example of how easily bioweapons can be spread and how it affects other people. So the Novichok poisoning occurs when individuals are exposed to the nerve agent, which are a group of highly toxic chemical substances. These agents can enter the body through various routes, and their effects are rapid and severe. So typically, this is how the poisoning happens. Inhalation. So the agents are inhaled as a vapor or aerosol, and if they're released into the air, people in the vicinity can breathe in the toxic fumes, leading to rapid absorption through the respiratory system. By skin contact. So contact with the skin can also result in absorption of these agents. This can occur if the agent is present on contaminated surfaces or objects. Skin exposure can lead to localized symptoms and systemic effects if a sufficient amount is absorbed. Ingestion, so swallowing or ingesting fluid, so liquids, food contaminated with agents can lead to poisoning. This route is less common, but it can occur if the agents are deliberately introduced into food or beverages. And by injection, so deliberate injection of an agent can lead to poisoning. It's uncommon as a route of exposure and typically requires a deliberate administration of the agent itself. But these agents exert their toxic effects by inhibiting the action of enzymes known as acetylcholinesterase, which are essential for nerve function. I had to write that one down to remember it. When these enzymes are inhibited, there is an accumulation of the neurotransmitter acetylcholine, leading to overstimulation of nerves and disruption of normal bodily functions. This then results in symptoms such as muscle weakness, twitching, paralysis, respiratory failure, and death if it's not treated promptly with antidotes. And it's important to note that Novichok agents are classified as weapons of mass destruction, and their use is prohibited under international law. Any suspected exposure to Novichok or other nerve agents requires immediate medical attention, and specialized antidotes and supportive care are necessary for treatment. Additionally, safety measures and protective equipment are crucial when responding to incidents involving these agents to prevent further exposure. And unfortunately, again, in this particular incident in Salisbury, we had responders who then got sick as a result of being exposed to the person or the people that had had the initial poisoning. So again, whilst we could say this is a chemical type or related terrorist event, we have to understand that anyone who's contaminated then becomes a biological threat to others, because we can get a spread of whatever that substance is, and obviously then spreading of any conditions that those people suffer as a consequence of the exposure. So what is the ideal biological weapon? Well, what we want, or we certainly need, is a high potential for person-to-person spread. We need high morbidity and mortality, but what we do need is a low infective dose. So we only want a small amount that can infect a high number of people, and most certainly infective by aerosol as a means of delivery. We want a lack of rapid diagnostic capability. In other words, it's difficult to detect, to try and understand and work out what has caused the symptoms or the problems. Lack of universally available effective vaccine. Potential to cause anxiety, and I mentioned about fear earlier on. And if we have a mass hysteria or panic due to this, that causes problems in itself. Availability of a pathogen and feasibility of production. In other words, whoever has the intent of doing something bad needs to be able to get hold of this easily, and needs to be able to produce or increase the amount that they have so that they can exert the biggest effect. We need also environmental stability with the potential for it to be weaponized. So if we were to get an exposure, we're going to be looking for clues. And as emergency responders and medical professionals, it's important for us to be able to recognize those and diagnose the problem. So here we've got a few epidemiological clues to a bioterrorism event. So we've got a large number of ill persons with similar disease or syndromes that present on or around the same time. We've got failure of common diseases to respond to therapies that we may initiate. We've got a higher mortality or morbidity than we would normally expect with a common disease or syndrome. An unusual, atypical, or genetically engineered strain of an agent. Disease with unusual geographic or seasonal distribution, and similar genetic type among agents, isolated temporarily or spatially distinct sources. So I also mentioned biosafety levels. And courtesy of our friends at the Center for Diseases Control, if you take a screenshot of this and you can copy the link that I provided here, this is a quick learn lesson. It takes around 15 minutes to complete, and it will give you the ability to be able to recognize characteristics of the 4 biological safety levels that we currently have. So biosafety levels, they're often abbreviated as BSL, are a set of standardized containment levels used to classify and describe the specific safety precautions and laboratory practices necessary when working with various biological agents. And that includes us as responders. These levels are crucial for ensuring the safe handling and research of potentially hazardous biological materials. And in the context of bioterrorism, understanding and implementing biosafety levels are essential to prevent accidental or intentional release of dangerous pathogens. So there are 4 primary biosafety levels, each with increasing levels of containment and safety measures. So our BSL-1 is our lowest safety level. So basically, we've got pathogens or substances that are not known to cause disease in the adult human. But level 2, we have a moderate danger due to inhalation, swallowing, or exposure to skin. And an example here would be influenza. BSL level 3 is potentially lethal disease, so severe. And we'd be looking at HIV, H5N1, and again, influenza. Our highest level, so BSL level 4, is life-threatening disease. So we have disease like Ebola, SARS-CoV-2, COVID-19, as we currently know it, would be on that level. So biological agents in the context of bioterrorism refer to living organisms or substances derived from living organisms that can be used to cause harm, illness, or death when intentionally released or disseminated. So these agents are typically used as weapons in acts of bioterrorism. So they can include various microorganisms, toxins, or infective agents that have got the potential to threaten public safety and health. So these are characterized based on their ability to cause disease and severity of the illness that they can produce. So some examples would be bacteria, viruses, fungi, and toxins, such as ricin and botulinum toxin. And it's important to note that biological agents can be naturally occurring pathogens or genetically engineered to enhance their virulence or resistance to treatment. So due to their potential for widespread harm and the challenges they pose in terms of detection and response, the use of biological agents as weapons is a significant concern for national security and public health authorities. And again, we have categories of what we describe as critical biological agents, as described by the CDC. So for category A, we've got our high mortality, category B, a moderate mortality, and category C, those that are easily produced. As you can see there on the table that we've got on the slide, we have examples there of the various type pathogens that could be involved. And what I'm going to do now in the next few slides is go through each of those categories and list some of those examples. So for our category A agents, these are the highest priority critical biological agents, often referred to as category A, characterized by their extreme potential for harm, as well as their ease of dissemination. These examples include anthrax, smallpox. I'm going to talk a little bit more about smallpox in later slides, the plague, and the botulinum toxin. Our category B agents. So these agents are considered to be moderately critical. So whilst they might not be as lethal as the category A, they can still cause significant harm and a public health threat. So we've got brucellosis, Q fever, and viral encephalitis agents. Category C agents are considered emerging pathogens, and they have the potential to be used as biological weapons in the future. So these could include Nipah hantavirus, and also multi-drug resistance tuberculosis. These themselves may require additional research and surveillance due to their potential threat. So the classification of critical biological agents helps guide government agencies, public health officials, and law enforcement in prioritizing their efforts for preparedness, response, surveillance, and research. Preventing the unauthorized access, use, or release of these agents is of utmost importance for national security and public safety. So if we look at probable bioweapons, in terms of the agents that could be used for bioterrorism, are typically those that are relatively easy to obtain, ideally naturally occurring, that can be collected quickly and easily. Easy to manufacture, as well as disseminate, as well as those that cause significant harm. And it's important to note that discussions about bioweapons are speculative, and actual use of such weapons is of grave international concern. It's not a matter of if, it's when. We know that we have the capabilities of producing such things as weapons, and if we do as legitimate agencies, then we can only imagine what illegitimate agencies may wish to do in terms of causing harm. Some of the agents that are considered more probable bioweapons due to their characteristics include, but are not limited to, anthrax. I've mentioned anthrax already. They can survive for an extremely long time in the environment, making for them a concern as a bioweapon. And again, we know they've been used in the past. Inhalation anthrax in particular can be deadly if it's not treated and recognized promptly. Smallpox. So smallpox was eradicated through vaccination, but eradication does not mean it no longer exists. So we still have legal stockpiles of smallpox in laboratories in the US, in America, and in Russia. It is also known that there is an extremely high likelihood of storage in other countries as well. And intelligence services are wary of that. Like I said, it still exists in laboratories. Just because we use the term eradication doesn't mean it no longer exists. Therefore, it could still get into the local population if somebody was able to get hold of it, or it was unintentionally released, or maybe intentionally released. It has the potential for use as a bioweapon because of its high mortality rate and its contagiousness. The botulinum toxin is one of the most toxic substances known. In even small amounts can cause paralysis and death. Botulinum can be synthesized and potentially used as a bioweapon. If you go to the CDC webpage, you can actually do a search for botulism, and there's quite a large amount of information there that you can read. Also the same for ricin. You can go to the CDC, webpage. Ricin is a toxin derived from castor beans, relatively easy to obtain. I'm sure most, if not all of you, have heard of castor oil. And even small amounts can be lethal if ingested, inhaled, or injected. Please again take the opportunity to take screenshots of these slides. So, tularemia. This bacterium can cause a severe and often fatal disease. It's highly infectious and has been studied as a potential bioweapon. So tularemia is a disease that can infect animals and people. So rabbits, hares, and rodents are especially susceptible and often die in large numbers during outbreaks. People can become infected in several ways, and that includes tick and deer fly bites, skin contact with infected animals, drinking contaminated water, inhaling contaminated aerosols, or agriculture and landscaping dust. So potentially we could have a risk of laboratory exposure as well. And in addition, people could be exposed as a result of bioterrorism. So we also have our viral hemorrhagic fevers. So viruses like Ebola and Marburg can cause severe hemorrhagic fevers with high mortality rates. Whilst not easy to weaponize, their potential for mass casualties makes the concern. So the Ebola and Marburg viruses are the most likely of viral hemorrhagic fevers to be used. And attempts have been made to use these viruses as weapons by aerosolizing infected body fluids or rodent excrement. So again, if you were able to get to an area where you have an Ebola or a Marburg outbreak, if you're able to collect biomaterial, there's the potential there to reproduce or certainly weaponize it in some way by putting it into diluting it into other liquid, aerosolizing, and then spreading elsewhere. We've also got plague. So the bacterium responsible for the plague can cause highly contagious and deadly infections, and that's also been used historically as a bioweapon. Again, I've got a good link there for you to follow. And we should also look at agricultural pathogens. So some bioweapons may target crops or livestock. So up to now, I've kind of talked about those that may infect people and animals, but we also need to consider food sources. This will disrupt food supplies and economies, and this can have a far-reaching effect. Examples might include foot and mouth disease and plant pathogens. So crops can be affected by biological weapons, including pathogens and toxins in various ways. And it's important to clarify that discussing this topic, again, does not endorse or encourage any harmful activities, purely for informational purposes. So pathogenic microorganisms like bacteria, viruses, and fungi can be used as biological weapons against crops. They may be engineered or selected to target specific plant species or varieties. So once introduced, these pathogens could cause diseases that spread rapidly amongst crops, leading to reduced yield, quality, or even complete crop failure. Toxins produced by certain microorganisms can also be used, and these toxins can be applied directly to crops or released into the environment. So it could be released into a water source, and that affects plant health and productivity. Some toxins are specific to certain plant species, making them effective tools for crop destruction. Biological control agents are also used as a means of getting rid of disease as well. And remember, we routinely use chemicals as a means of pesticides. In some cases, beneficial organisms used in agriculture, such as insects or nematodes, can be manipulated to harm crops instead of protecting them. So, for instance, parasitoid wasps or predatory insects might be modified to attack and damage crop plants. I'm sure everybody's heard of GMOs, so genetic modification. Advances in biotechnology have enabled the creation of genetically modified organisms, and we have genetically modified crops to hopefully make them more resistant to substances and the agents that could contaminate them and therefore affect growth and yield. So, for example, GMOs could be designed to produce substances toxic to plants or to disrupt their growth and development. We should also include chemical manipulation. So biological weapons can be combined with chemical agents to enhance their effectiveness. So again, we've called this a bioterrorist or bioterrorism, but we should also be aware that biological substances can also be mixed with chemical substances. These chemicals can be used to weaken plants' natural defenses, making them more susceptible to biological attacks. So it's crucial to emphasize that the use of biological agents as weapons is a serious crime under international law, and the global community has made efforts to prevent such things occurring. So there are various treaties, various conventions and regulations that are in place to combat bioterrorism and hopefully ensure the safe handling and containment of any dangerous pathogens. Efforts to strengthen biosecurity and counterterrorism continue to be a priority for governments and international organizations. So as an example, we've got the Biological and Toxin Weapons Convention. And what we need to understand is that we have biological research for peaceful purposes alongside international cooperation. So this particular convention is the cornerstone of international efforts to prevent disease from ever being used as a weapon. And Article X of the convention states that countries have the right to participate in the fullest possible exchange of equipment, materials, and scientific and technological information for the use of bacteriological agents and toxins for peaceful purposes, and that parties to the convention in a position to do so shall also cooperate in contributing individually or together with other states or international organizations to the further development and application of scientific discoveries in the field of bacteriology for the prevention of disease or for other peaceful purposes. And if anybody wants to refer to that convention, you can go to the eeaseuropa.eu webpage that will answer an awful lot of questions in terms of what are biological weapons. And this is a very important document that governs us in Europe. Unfortunately, the safe use of biotechnology does not stop it getting into the general population, as we've demonstrated in Wuhan in China. In this case, there is a huge argument against the manufacture, procurement, and development of any technology with the potential to cause worldwide biological harm. So the argument is that if it did not exist and was eradicated, then there would be no need to develop a cure. But unfortunately, we cannot trust other people to not attempt to weaponize anything. Smallpox, as I've already mentioned, whilst officially, according to the WHO and CDC, has been eliminated, there still remains stockpiles worldwide. For us, I think the most important thing is recognizing any kind of bioterrorist or bio incident and managing it. So managing a bioterrorism attack or an incident requires a coordinated and multifaceted response that's going to include various agencies, healthcare professionals at varying levels, and the public themselves. So what I'm going to look at here is a few key steps that we can use to best manage a terrorism attack. So early detection and reporting. So early detection of a bioterrorism incident is crucial. Healthcare providers and laboratories should be vigilant for unusual clusters of illness or symptoms and report them to public health authorities promptly. The public should also be educated on recognizing unusual symptoms and reporting them to healthcare professionals or local health departments. So again, one of the purpose of this presentation is education. Education for us or to us as healthcare professionals, so that we that can then in turn pass that on to our colleagues who can maybe watch this presentation back, which will then lead to further reading and further education. Not forgetting the public. They should be educated on recognizing unusual symptoms and reporting back and telling us. Isolation in quarantine is a key tool in terms of managing any incident. We obviously know that in terms of the recent pandemic and the need for isolation and quarantine to stop and limit any kind of spread. Now, if a bioterrorism agent is identified, individuals who have been exposed or infected should be isolated or quarantined to prevent further spread of the disease. Healthcare facilities and public health authorities must have plans in place for managing isolation and quarantine procedures. Much as we did on my oil rig here in the middle of the North Sea, when we had cases of COVID-19 on board, when they tested positive with me, they were quickly put into isolation, isolated away from the rest of the crew, and thereafter barrier nursed by me until such time as we could evacuate that person. And we were also able to put on a specialist flight to be able to maintain that level of isolation even when they reached shoreside, and they were then taken on to a hospital or other facility as needed. For us, again, as medical professionals, medical treatment is important to know about and understand, and that's not always going to be the same. It really does depend on the nature of the problem. And remember, when we're working through our C, A, B, C, D, E, etc., the level of interventions that we may wish to initiate under normal circumstances may not be possible in the event of an incident like this. It could actually expose us to further dangers as responders when trying to initiate those procedures. Patients exposed to these agents may require specialized medical treatment and supportive care. So it's essential to have adequate medical resources and, of course, trained personnel to be able to manage it. We also need stockpiles of medical countermeasures. So that's going to include things like vaccines, like antitoxins and antibiotics that need to be readily accessible for rapid distribution. We also need a good public health response. So public health agencies should lead the response efforts, including contact tracing, monitoring of exposed individuals, and epidemiological investigations. Most certainly for us, working in wilderness, rural, and remote locations, we don't necessarily have access to laboratories where testing and we can look at samples under microscopy. You know, these facilities may not be readily available to us. So we need access to that. I have the ability to be able to take samples in my location, but I can't test and I can't examine. Whilst we may have some form of rapid diagnostic test, it may not be applicable for the nature of the thing that we're trying to respond to and manage. Communication with the public should be transparent and it should be accurate, providing information on a situation, preventative measures that are necessary, and of course available resources, so where people may need to go to or the actions that they may need to take. As was my job when I was in the fire service, as part of the New Dimension initiative, decontamination of a large number of people may be necessary. So decontamination procedures should be established to treat exposed individuals and contaminated areas. You're going to need decontamination facilities, you'll need protocols and trained people readily available. It's the old adage, the solution to pollution is dilution. But we also have to consider, where does the dilution go? What we don't want is that going into natural water sources where it can spread even further. So do not forget to consider where the diluent or runoff goes. Where we have law enforcement and security measures, we need to understand that law enforcement agencies play a critical role in investigating incidents, identifying perpetrators, and ensuring public safety. Remember, I said it's a multifaceted approach. We cannot do this on our own, and we do need to get other individuals involved, but at the same time, we don't want other individuals getting contaminated and any kind of contaminant spreading. So security measures should be implemented to protect potential targets of bioterrorist attacks, such as critical infrastructure and research facilities. So as remote healthcare providers, we cannot manage this on our own, as I've just said. So now is the time to ask for help. We must have command and control. So what we need to do is know and understand local potential threats. So for me, working offshore in the middle of, in the North Sea, you would think, how on earth is anybody going to be able to get to me? Well, actually, it's been a concern for oil and gas installations worldwide for a significant number of years. So that's somebody who could potentially come to us. We actually have a guard vessel that looks after us. So if there was any kind of fishing boat, any kind of vessel that tried to get inside our restricted zone, so we have a 500-meter restricted zone, anyone that tried to get into that could potentially be a threat to the installation. Also, on board the installation, we have a number of people. We do not always have the same people all the time. We consistently get vendors and new people that might come out to do specialist tasks. And, if we don't know them, if we don't vet them, and, there is potential. They say where there's a will, there's a way, that somebody could bring something with them that could be in a sealed container that could get released on board. But at the same time, we have substances that we use for oil and gas production that could also be a biological hazard. So we have things called biocides. Now, in oil and gas production, especially in our oil pipelines, we get naturally occurring bacterial growth. And we use biocides or biocide chemicals to kill. So essentially, we have a biological agent that fights against other biological agents to kill it and get rid of it. But that could be spilled if it's not contained correctly, which means potentially it could be exposed to other people. So we actually have, as part of our emergency response on board, procedures to be able to manage things like spillages of such an agent. And if we're going to put it into our production system, there are very strict measures that we must follow, most certainly in terms of personal protective equipment and protecting ourselves, as well as risk assessment and written procedures that we have to follow. So coordination and collaboration. Effective coordination among local, state and federal agencies, as well as international partners, is essential for a successful response. Interagency drills, training exercises, and communication protocols should be established in advance. And most certainly, when I was in the fire service, this is something we trained on a regular basis. There were a large number of vehicles and personnel that could be called out at any time. In fact, because we were specialist trained, you couldn't always guarantee that we would be on duty, in which case we would carry individual pages with us. and we could literally be called at any period of time during your duty period, even when you were at home, to respond to an incident. So for myself, because I was one of the operators, I actually drove one of the large vehicles that we had that contained all of the decontamination equipment. So not only did I drive that, I operated the vehicle itself. It had various kind of mechanical things that it could do, which would help us set up the equipment. So I was also the vehicle commander for that as well. It's important that you train, and it's important that you work with other agencies so you all know what you're doing. The last thing you want to be doing this is for the first time with somebody you've never met before. And we need community preparedness. So communities should be prepared for bioterrorism incidents by developing and practicing response plans. Public education campaigns should promote awareness, preparedness, and reporting of suspicious activities. Research and development. Ongoing research into vaccines, treatments, and diagnostic tools for potential bioterrorism agents is crucial. Collaborative efforts between governments, academic institutions, and pharmaceutical companies can advance preparedness efforts. So for legal and ethical considerations, laws and regulations regarding bioterrorism response, biosecurity, and biosafety should be in place and enforced. Ethical considerations related to research, surveillance, and response must also be addressed. Managing a bioterrorism attack is going to be a complex and challenging task. But if we are well prepared and we can ensure a coordinated response, we can minimize harm and protect public health and safety. So these preparedness efforts that need to include regular drills, training exercises, are essential for us to ensure an effective response. There is text available to you that is available in terms of managing bioterrorism. In fact, I have from my colleagues at the OPCW, so the Organization for the Prohibition of Chemical Weapons, a text here on bioterrorism preparedness, attack, and response that includes decontamination procedures. So you can go to the CDC website, the OPCW webpage, and you can download that information there if it's something that's new to you and you need to introduce that to the agency that you work with, or for your personal knowledge. The OPCW webpage is a really big webpage. It's really good. There's a lot of information there, and it is worth spending some time going through that. Again, worthy of a screenshot, so you can get the link there and have a look. So in summary, bioterrorism involves the deliberate use of biological agents to cause harm, illness, or fear. This threat has implications for remote or wilderness healthcare practitioners who work in isolated or natural settings. So these practitioners must consider the following. Risk assessment. We should be aware of the potential for bioterrorism in our areas, such as the release of harmful agents into the environment. They should stay informed about local and global security threats. Again, another reason for us giving this presentation today. In terms of preparedness, training and resources should be available to recognize and respond to unusual illnesses or symptoms that may result from a bioterrorism incident. Communication. Establishing effective communication networks with local agencies, so public health agencies and emergency responders, is crucial for timely information exchange and coordination during the bioterrorism incident itself or the event. Protection. Practitioners should have access to personal protective equipment, so PPE, and protocols for minimizing exposure to potential biological threats. So right by my door here, as I exit my sick bay, I actually have a safe PPE for staff. There's a poster on the door there, and depending on the nature of the incident I'm responding to, then I need to select the appropriate PPE before I leave. Hopefully, I've also got a good idea of what I am responding to, and if there is any physical, or any chemical or biological threat, I will be informed of that before I respond. Healthcare workers in remote areas should know how to report suspicious activities or symptoms that could be related to bioterrorism to the appropriate authorities. So again, in the case of infective disease, I have telephone numbers behind me on the wall that I can also get in contact with. Finally, community education. Promoting awareness and education among the local population about the bioterrorism risk and preventative measures can enhance our overall community safety. In remote or wilderness healthcare settings, practitioners may face unique challenges in responding to a bioterrorism threat due to the limited resources that we have available to us, as well as our isolation. Therefore, being well informed, prepared, and connected with broader healthcare and security networks is essential for us to effectively manage such a situation. So here I've provided some resources for you to be able to go away and read further. Like I said, this is a weighty topic. In fact, it's been quite challenging for me within the limits of around 45 to 50 minutes of put together a presentation that, one, makes sense, but at the same time provides you with enough information so that you can go away and you can go and research a little further. Bioterrorism and the role of the clinical microbiology laboratory. That's a pretty interesting article there. It describes, or it details information that relates to laboratory use of such pathogens and how to minimize things like release. This gives us an idea of potentially how things could get released into the public. But it also details things like appropriate countermeasures to stop this from happening. And it might be that you may work in such an environment. UN Counterterrorism Implementation Task Force, again provided through the OPCW webpage, and it describes interagency coordination in the event of a terrorist attack. Like I said, the OPCW do have quite a lot of online and learning resources that you could go to. I mentioned there about smallpox virus stocks. There's some quite interesting information there. It's also interesting to note that really this is nothing new for us. In fact, during the 1950s in the US military was carrying out essentially clinical trials by purposely spreading biological pathogens in New York City. If I remember, Operation Big City was the name of the project, and that's an interesting one to look up, to understand how things could be easily disseminated and delivered into a local populace. The Biological Weapons Convention I discussed earlier, there's more information there. And as well as it affecting people and animals, we should also consider crops as well, because of the longer term and the far outreaching effect that can have on a populace. Whilst not directly affected themselves, if the environment is affected, there is a knock-on effect that could be significant. So I'm going to open the floor up to any questions. And again, if you guys have any questions about the presentation that I've given today, please do not hesitate to email us at courses@corom.org, and we will do our very best to get back to you. Also, you can email me on t.cranton@corom.edu.mt, and again, I will do my best to provide an answer to your question. And again, we have to understand that this is nothing new. We've got Cortez here. When he first stepped foot in what we know as Mexico, they took disease with them, and that disease rapidly spread amongst the local population, who were not used to being exposed to that level of diseases. Now, you know, you can argue here that this was an unintentional release, but this is a good example of how pathogens or things that you know could potentially be used as bioweapons could easily spread and affect a community and a populace. Again, thank you very much for your time, and I look forward to the next time that I have a presentation for you. Thank you.